In the third week of development, the events taking place are differentiation of ectoderm in three functional zones. The ectoderm differentiates into surface ectoderm, which gives rise to epidermis, neuroectoderm, which gives rise to central nervous system, and the pluripotent cells will give rise to the primitive streak. The primitive streak is formed at 15th day of intrauterine life. It acts as a primary organizer and it helps in the formation, it induces the formation of notochord and formation of intraembryonic mesoderm. Once the intraembryonic mesoderm is formed, the bilaminar germ disc is converted in trilaminar germ disc and the process is called gastrulation. The intraembryonic mesoderm will not enter in between the epiblast and hypoblast in two areas, buccopharyngeal membrane and cloacal membrane. And at the most cephalic part, we have the formation of pericardial bar, and allentoenteric diverticulum will develop from the yolk sac and it will enter into the connecting stalk to vascularize the chorion. And the changes taking place in the trophoblast is formation of tertiary villi and branching villi. Most significant change occurring at the third week of intrauterine life is the formation of trilamellar germ disc. It is also known as gastrulation. So, with the advent of intraembryonic mesoderm, the bilaminar disc is now converted to a trilaminar germ disc. So, suppose now here we have the amniotic cavity, this is the germ disc, and this is the yolk sac. So if we remove the amniotic cavity and if we see the germ disc from above, this will be the appearance. So initially the germ disc is circular, then it gets elongated and then it becomes pear shaped. It is broad in the cephalic part and narrow in the caudal part. So at 18th day of intrauterine life, it becomes pear shaped. Now the surface, this ectoderm will differentiate in three functional zones. So there is surface ectoderm. So this is the surface ectoderm which will then develop or it will form the epidermis. In the midline, a concavity or groove is there. It has neuroectoderm and it will form neural tube and the central nervous system develops from it. And caudal, in the caudal part we have the pluripotent stem cells. So at 15th day of intrauterine life, the primitive streak is formed from the pluripotent cells. It will move cephalically and towards the cephalic part a knob like structure is seen. It is called primitive knot. In the primitive knot a pit develops known as blastopore or primitive pit. So the primitive streak is the primary organizer because it induces formation of notochord as well as the formation of intraembryonic mesoderm. So the intraembryonic mesoderm and the notochord formation are induced by the primitive streak. Also, the cells proliferating from the primitive streak will replace the primitive ectoderm and endoderm into definitive ectoderm and definitive endoderm. Thus, all the three germ layers are derived from the pluripotent cells arising or proliferating from the primitive streak. So, the primitive streak will disappear by about 26th day. Now, the cells proliferating from primitive streak, when they proliferate cephalically in the midline up to the procaudal plate, it gives rise to the notochord. Means from the primitive knot in cephalic direction up to the procaudal plate in the midline, it gives rise to notochord. So this notochord is again present between epiblast and hypoblast, and rest of the cells are proliferating and they will proliferate and they will occupy the space between the epiblast and hypoblast. As these cells are present within the germ disk or within the embryo, these are known as intraembryonic mesodermal cells. So with the advent of intraembryonic mesoderm, there is formation of trilamellar germ disk. It is known as gastrulation. Okay, so this intraembryonic mesoderm is migrating between the epiblast and hypoblast except at two places. So in the cephalic part we have the procaudal plate where only epiblast and hypoblast are firmly adherent and there is no intraembryonic mesoderm, and it will form the buccopharyngeal membrane which will rupture at about fourth week, and the oral cavity will communicate with the alimentary tract as it is present in the floor of stomatodium. Then in the caudal part we have cloacal membrane which again has epiblast and hypoblast. It does not have intraembryonic mesoderm, and the cloacal membrane will form the anal membrane and the urogenital diaphragm and the urogenital membrane. So both this cloacal membrane will rupture at about 7th week, and if we see the structure of the disc now, there is procaudal plate, then in between the epiblast and hypoblast we have the intraembryonic mesoderm. In the central part there is neuroectoderm. From the primitive knot to the procaudal plate we have the developing notochord. This is the primitive strip caudally present just above the cloacal membrane and connecting stalk. So this is the cloacal membrane and this one is the connecting stalk. So what are the major events taking place? There is appearance of primitive streak. It will induce formation of notochord and intraembryonic mesoderm, and later on gastrulation means conversion of bilaminar disc into trilaminar disc occurs. Now here we can see the derivatives of ectoderm. Mainly from the surface ectoderm, the epidermis will develop. From neuroectoderm, central nervous system will develop. And then the mesoderm gives rise to blood, lymph, bones, and the musculature of excretory and reproductive system. Endoderm will line the alimentary tract, respiratory tract, and the excretory and reproductive system. The primitive streak will induce formation of notochord. So we can see the primitive streak at the cephalic part forms primitive knot in which a pit develops, and this pit is known as blastopore or primitive pit. So this is the blastopore. Now from here the cells from the primitive streak will start migrating in midline cephalically up to the procaudal plate. So in the cephalic direction, the cells will migrate from the primitive streak up to the procaudal plate, and this is known as notochordal process. So initially it is a solid cord and later on it gets canalized to form notochordal canal. Okay, so initially it is a solid process and it is known as notochordal process. Then it gets canalized and it will form notochordal canal. Now the cells of the notochordal canal will fuse with the cells of the endoderm, and both these cells will disappear, both these layers will disappear, right? So the layer of notochordal canal and the layer of endoderm will fuse and it will disappear. So what will happen? There will be communication between the amniotic cavity and and the yolk sac. Okay, so communication between the amniotic cavity and yolk sac is possible because of a canal known as neurenteric canal. So this neurenteric canal is formed so that the nutrition from the yolk sac will enter in the amniotic cavity and it will supply the ectoderm. After that, the cells of the notochord will intercalate with the endodermal cells. A groove is formed, notochordal groove. The ends will fuse. Then the notochord will exhalate itself from the endoderm, and this endoderm which disappears, it is now replaced by the cells proliferating from primitive streak, and now a definitive endoderm is formed, and this notochord present in the midline is now known as definitive notochord. So initially we have the notochordal process extending from primitive streak to prochordal plate. It gets canalized to form notochordal canal. There is fusion of the cells of the notochordal canal with the endodermal cells. It will disappear to form the neuroenteric canal. After that, the uh, the cells of the notochord, they will form notochordal plate. Okay, this notochordal plate will form a folding groove, it will form a groove, and the ends will meet, and now the definitive notochord is formed in the midline. So in this slide we can see this is the epiblast, definitive epiblast. Then this is all intraembryonic mesoderm on either side of the notochord. In the midline we have the notochord, and on either side of the notochord is the intraembryonic mesoderm, and this is the definitive endoderm. Now, the notochord acts as a secondary organizer. It will induce the formation of neural tube from which the central nervous system develops. Why we need notochord? Actually, notochord acts as a guide, or it acts as a mold for formation of vertebral column, and once the vertebral column is formed, the notochord will disappear and it will leave its two remnants. So the nucleus pulposus present in the intervertebral disc and the apical ligament of the dens are the two remnants of the notochord. Now, allantoic diverticulum will arise from the yolk sac. So this is the wall of the yolk sac. A projection appears along its caudal part. It is known as allantoenteric diverticulum, and this will enter into the connecting stalk, which is going to form the future umbilical cord. So this allantoenteric diverticulum will vascularize the connecting stalk, and it will give rise to the umbilical blood vessels, and it will then form urachus. The urachus extends from the tip of, or from the apex of the urinary bladder up to the umbilicus, and later on the urachus will obliterate and it will form median umbilical ligament in the adults. So the remnant of the urachus is median umbilical ligament, which is seen in the midline infraumbilical part. Okay, now now at the 21st day, how the embryonic disc appears. So we have removed the ectoderm. Okay, so after removing the ectoderm, we can see the surface ectoderm is there, which will give rise to epidermis. The new ectoderm will form the vesicles, so it will give rise to the central nervous system. Most cephalically, the horseshoe shaped area is known as it is known as cardiogenic area. So this is the cardiogenic area. It is also known as pericardial bar. Then we have the procaudal plate, where only two layers of the germ disc are there, epiblast and hypoblast is there. It is not um, means it is not advented by the intraembryonic mesoderm. So it will form the buccopharyngeal membrane, which on rupture at fourth week will develop the oral cavity. Then after, now if we see in the midline, first we have the pericardial bar, then we have the procaudal plate which forms the buccopharyngeal membrane, then the new rectoderm gives rise to the central nervous system, then we have the primitive knot, primitive streak, and this is the cloacal membrane, where again only epiblast and hypoblast is there, it will rupture at 7th week. It forms the anal membrane and urogenital membrane, and most caudally we have the connecting stalk from which the umbilical cord will develop. So this is the structure of embryonic disc at 21st day. So far we have seen the changes in the embryoblast. Okay, now we will see the changes in trophoblast. So up to the 2nd week of development, primary villus are formed. So how the primary villus is formed in the trophoblast? The cytotrophoblast present in the core part and surrounded by the syncytial trophoblast is known as primary villus, and primary villus will develop at late 2nd week. Now how the secondary villus develops? So in the inner core we have the extra embryonic mesoderm. Okay, then it is surrounded by cytotrophoblast, and the outermost layer is the syncytiotrophoblast. So when these three layers are there, it is known as secondary villus, and it is appearing at the beginning of the 3rd week. Later on, tertiary villus or true villus will develop. So in the central core, we have the fetal blood vessels which are arising from the allantoic, coming from or projecting from the yolk sac in the connecting stalk. So it will develop the fetal vessels. Then we have the extraembryonic mesoderm. Then there is cytotrophoblast, and then there is syncytiotrophoblast. So at the 3rd week of development, there is formation of tertiary villus. Also, branching villi will develop. Now see, this is the tertiary villus. Now what are the anchoring villi? So this is the truncus, then ramus and the ramulus. The branches of villi will project in the intervillous space. Now see, this is the maternal side which is formed by decidua basalis, and the blood vessels or the blood from the maternal side is entering in the intervillous space. So this is the intervillous space in which maternal blood is there. In between the intervillous space we have the villus. They can be primary, secondary or tertiary. So anchoring villi are there, and these villi are arising from the chorionic frondosome. So chorion is the fetal side and the deciduous basalis is the maternal side, and in between these two plates we have the anchoring villus formed by the fetal side, and intervillous space has maternal blood. So these changes occur in the trophoblast up to the late 3rd week of development. Now we will see the applied aspects of developmental anomalies in the 3rd week. So normally the primitive streak appears at 15th day, and it induces the formation of notochord, intraembryonic mesoderm. It forms definitive notochord, definitive ectoderm, definitive endoderm, and then it disappears at 26th day of intrauterine life. So the primitive streak should normally disappear at 26th day of intrauterine life. But if it persists as sacrococcygeal teratoma, then the teratoma is benign. It is common in females, seen in sacral region, as it is derived from the pluripotent stem cells of the primitive streak. All the three germ layer derivatives are seen, like hair, bone, cartilage, muscles. These all are seen in the sacrococcygeal teratoma. It has good prognosis and it can be removed surgically. And there are four types of sacrococcygeal teratoma. First, when it is type 1 is entirely outside the pelvis. Type 2 is mostly out of the pelvis. Type 3 is mostly inside the pelvis, and type 4 is entirely inside the pelvis. Then there are some teratogenic agents like radiations, infections, or X-rays. So if these if the fetus is exposed to these teratogenic agents between 15th to 18th day of development, it results in malformations. Okay, now the next slide or diagram you can see is of the urachus. Okay, so normally at 16th day the allentoenteric diverticulum appears as an outpouching of the yolk sac, and it enters into the connecting stalk, and then the allantois form urachus. Urachus extends from the apex of urinary bladder to umbilicus. After birth it will obliterate to form median umbilical ligament. Okay, and it forms blood vessels. When it enters into the connecting stalk, it forms umbilical vessel. It vascularizes the chorion. Now, normally the allantoenteric diverticulum should get obliterated, but if it does not obliterate and if it persists, then it results in urachal cyst or sinus or fistula. So what is urachal cyst? It is the remnant of lining epithelium of the urachus. Okay, so in the C you can see there is urachal sinus, B is urachal cyst, and A is urachal fistula. So in urachal cyst, the proximal and distal part is obliterated, and only in the middle part you can see the persistence of urachus. So it is called urachal cyst. In urachal sinus, it is the remnant of urachus that communicates with the bladder or outside the body. Okay, so one end is over the umbilicus, other end is to the bladder. So half of it will get obliterated and half of the part will either communicate with the bladder or outside the body through umbilicus. Then the third is urachal fistula. It is the persistence of complete urachus. So it forms a passage from bladder to the umbilicus. So urination may occur through the umbilicus, that is urachal fistula. In the females who have high alcohol consumption during gestational period, mainly in the first trimester or the period of gastrulation, then it will result in holoprosencephaly. There are three types of holoprosencephaly: lobar, semilobar, and alobar. So in this, there is a small forebrain, and there are fused lateral ventricles, and there is hypotelorism. Telorism, hypo means the eyes are closely set. Okay, the eyes are close to each other. So it is called holoprosencephaly. And the second diagram is of cyrenomelia. It is called caudal dysgenesis. It is also known as mermaid syndrome. So as the mesoderm in the caudal portion of germ disc is insufficient, it will lead to the fusion of lower limbs, and it is also associated with the anomalies of vertebrae, then kidneys, genital organ, and there is imperforate anus. So this condition is known as cyrenomelia, and the reason is insufficient mesoderm in the caudal portion of germ disc.